Hello, my name is Mallory Bow, and this is my presentation on apoptosis as read from our last literature review. So apoptosis at the most basic level is cell suicide. It is a conserved eukaryotic cellular organismal function that works to remove excess or damaged cells that occur during development, regulation of the immune system, or from just general stress. If there is excessive apoptosis, this could be an indication of a degenerative disease. And if apoptosis becomes blocked, this could be an indication of cancer or remission. There are two pathways that can cause the activation of apoptosis, and it is shown here as either an internal mechanism on the right or an extrinsic mechanism or pathway shown here on the left, both of which result in cellular death. Anastasis is another important concept that we will be discussing during this presentation, and it is the name given to cells who are able to save themselves from apoptosis or murder. It is, again, a highly regulated process that we don't know a lot about, as we thought that once the molecule caspase had been activated, this was the point of no return, and the cell was dead. This is no longer the thinking, and we are now starting to see that some cells can actually save themselves. In the research article that I read, A Molecular Signature for Anastasis, Recovery from the Brink of Apoptotic Cell Death, the researchers were looking to determine two things. One, they wanted to find similarities between the genes, pathways, and cell behaviors of anastasis and wound healing. And then two, they wanted to identify several potential targets for therapeutic manipulation, i.e., cancer treatments. The implications of this research not only will aid our understanding of just generally how apoptosis works and anastasis, but how do those cells which we thought were dead seemingly come back to life? This research can be used to understand how to limit permanent damage to tissues in the cases of neurodegenerative diseases, and how some cancers can come back or potential pathways for some cancer treatments. So to conduct their research, the authors used HeLa cells, which were exposed to 3 hours of ethyl alcohol treatment, which causes significant cell shrinkage and blebbing, or the beginnings of apoptosis. This 3-hour treatment caused activation of caspase 3, 7, and 9, and cleavage of PARP1, and the release of cytochrome C from the mitochondria into the cytoplasm. This means that ETOH, ethyl alcohol, activates the intrinsic apoptotic pathway. On the left here, you can see a list of the various assays that were performed on the HeLa cells. In this first video, you will see that approximately 70% of the cells attach to the matrix, and they begin to spread out again. So we thought that these cells were going to be dead. That's where they're little and they're tiny. But then, that's after the ethyl alcohol treatment. If you notice the timestamp up here, this is hours. They start to come back. They look different, but they are in fact living and proliferating again. In the second video here, and this is actual research from the study, you can see the activation of the caspase 3 in the green. So in this fluorescent green here, and it occurs in about 75% of these cells. So within a couple of hours, that caspase 3 has been activated. And by the time we reach 6 hours, it is in about 75% of the cells. So still photos of both of these processes if you want to see individual time-lapsed shots can be found in the literature. You're going to want to look at figure 1. I thought the videos were much more interesting as they showed the live pictures. So as they performed their tests, one of the major things that the researchers did after they damage to the cells, and then they studied the events that happened afterwards, is they performed an RNA sequence. So they sequenced the RNA that came out of the cells. They used a kit to perform their RNA-seq, and then right here over on the left in diagram O, you can follow the pathway that they took. They took away the ribosomal RNA, compared it with a cDNA library, and then went through and they did use some software in all of their analysis of everything. And they, of course, did some PCR testing. And they found that there were certain genes that showed up commonly, so we're going to discuss those later. And they also found that there were really two levels of recovery. There was some early stage recovery mechanisms that happened, and then there were some late stage recovery mechanisms that would come into play, all of which led to the cell fully recovering, re-entering the cell cycle, proliferating, and carrying on with its cellular function, wherever that may be in the body. So the next test that the researchers performed after their RNA-seq was they decided to do a comparison just between those early and those late stage genes using a program called AutoSome. So this program is going to compare genes that are clustered together according to the similarities in their expression patterns. The most common genes found in the early 1 to 4 hour tests versus the 8 to 12 hour recovery cells are listed here. You've got your earlies and then your lates. The study results are also presented on the side in Figure A. This is how the various genes were determined. You can see this cluster here in diagram A came first, and that is during recovery hours 1, 2, 3, and 4. And then later in 8 and 12, they had quite a bit of similarity between a couple of genes that were found. Specifically, there are 1,172 genes that were discovered in here, and there were 759 genes down here, these late genes, that were discovered. And they are further elucidated over here in Figure B. So next, there was a comparison of these genes to the Kyoto Encyclopedia of Genes and Genomes database, which is a database for these. And the pathways that the genes coded for at each stage are listed here in the form of their data. So you can see the early pathways, endocytosis, MAPK, protein processing. Most specifically, they were interested in this TGF-beta signaling and then the cell cycle entry. In the late stage genes, they were very interested in this ribosomal biogenesis in eukaryotes, and you can see that it is significantly upticked here in the late response genes. So two of the specific pathways in the late response test were focal adhesion and regulation of actin cytoskeleton. Therefore, the researchers hypothesized that during the proliferation stage after 8 hours, that a cell might start to become migratory. So to test this, they scratched monolayers of cells treated with the ETOH and allowed them to recover for up to 16 hours. And they found when they did this that the treated cells did in fact recover faster and that their cell shape was elongated, indicating a slight amount of migration. And this increased shape change might be what is facilitating the migration and then the wound closures. This was a scratch test. So the results are shown here in the picture. We have the ETOH treatment, and we have mock treatment or the control. So you can see at 0, the tests are the same, and 13 hours later, you do have the closure of the wound here, where without the initiation of that apoptosis recovery or anastasis, the cell is still missing a piece of closure to it. And then if you look down below, you can see, indicated by the yellow arrows, that there is some change to cell shape. To determine which of the response genes induced during early recovery from the ETOH treatment were common in other recovery pathways, as one of the initially outlined research questions, they looked at 69 genes. And they ranked those against H. H4, which are human glioma cells. And the cells were tested in two different mediums, as both the H4 treated cells and HeLa cells will activate caspase-3, which was going to result in cleavage of PARP1. So the H4 cells were treated with ETOH. They already had an analysis of HeLa cells treated with ETOH. Then they additionally treated some HeLa cells with this STS, and this is the STS molecule here, and that is the full name for it. So STS is a protein kinase inhibitor, which means that it will induce the intrinsic apoptotic pathway, which can be seen in that cytochrome release, the cytochrome C release from the mitochondria into the cytoplasm, that PARP1 cleavage, and the caspase-9 activation. So inhibiting this pathway should block the STS-induced cell death. The researchers found that 63 of the 69 genes were upregulated in the H4 cells. And 64% of the ETOH-treated HeLa cells, comparatively, were able to recover. So those two became very similar for them. And you can see that over here as you look in Figure C. So in Figure C, the HeLa cells recovered treated with the ETOH. You can see the number of these genes as coded by the mRNA that was discovered were upregulated. You can see the number of H4 cells treated in the same medium, the ethyl alcohol, and there were a few that were downregulated and a few genes that were unchanged in the level of the production of these 69 genes. And then the HeLa STS treated cells, you can see that there is a change in them. There's a number of genes that have now been downregulated and a number of genes that have been unchanged. So what they figured out is the genes that were recovered were responsible for the regulation of the following mechanisms. Those are all listed down here. And this is similar to what they found when the HeLa cells finally recovered from the ETOH. They were able to find a comparable production of these genes after recovery. So cells that are able to save themselves, recovery from apoptotic stress, or anastasis, will ultimately re-enter the cell cycle. And the researchers wanted to determine which genes were expressed commonly across other types of growth and recovery. To do this, they looked at the expression of some top-ranked genes after the cell had gone through nutrition deprivation, which should induce cell growth and autophagy, which are for survival. And the researchers did not find that the expression of autophagy genes, therefore they were able to determine that autophagy and anastasis have two different mechanisms. By starving the HeLa cells of amino acids for 2 hours, the researchers were ultimately able to induce autophagy. So this is shown by staining the cells with an LC3. They were not, however, able to induce caspase-3 in that amount of time, which would lead to the cleavage of that PARP1. Finally, they did a gene comparison to further corroborate that the two pathways are in fact different. Next, the researchers noticed that there were transcripts corresponding to a subset of early genes that were already elevated in abundance in apoptotic cells compared to untreated cells. Therefore, the researchers had two hypotheses about why this might be. They proposed that there might be genes that apoptosis hadn't completely stopped, or that those genes were always at the ready in case apoptosis might cease.